All right, we're continuing at 27.2, which is actually not reflective of what the study guide had. But we're looking about how prokaryotes obtain nutrition, and there's 4 primary modes: photoautrophy, chemoautrophy, autotrophy, photoheterotrophy, and chemoheterotrophy. So these basically refer to how they obtain energy. A photoautotroph uses light and converts carbon into sugars and foods. So this is photosynthesis, if you like, very typical. A chemoautotroph is one that uses various chemicals to convert CO2 or a carbon source into energy. This is fairly a bit more common with prokaryotes, of course. And then heterotrophs, such as you, like a photoheterotroph, uses light to take organic compounds and convert them into energy or food. And then a chemoheterotroph, the same thing. Somebody who consumes chemicals, in this case, instead of using light, they consume chemicals and then use those chemicals to convert it into food. Animals, fungi, some prokaryotes, but all right, you get the idea. The 4 major modes. Prokaryotes all are affected in different ways by oxygen. Some like it, some don't. Obligate aerobes require oxygen. They're obligated to have air or oxygen, or they can't survive. These are the newer of the prokaryotes in the evolutionary time scale. As oxygen became more prevalent in the atmosphere, they became more and more well suited for the environment. Facultative anaerobes are kind of the in-between. They can survive with or without oxygen. They typically prefer one or the other environment, often with oxygen, but they can survive in either environment. And then obligate anaerobes are poisoned by oxygen. These are your not bacteria, but archaebacteria, various extremophiles such as thermophiles or methanogens, things like that, organisms that live in typically inhospitable environments that then they like to hide away from all the oxygen sources, and when there is oxygen around, they're poisoned and they don't like it. These are kind of the originators, the original prokaryotes, when oxygen was at very low levels in the atmosphere on early Earth. Some prokaryotes can metabolize nitrogen, of course, and we call this nitrogen fixation, where they take atmospheric nitrogen and convert it into ammonia, or NH3. Not all bacteria or prokaryotes do this. Nitrogen fixation is just one type of conversion of nitrogen, in this case atmospheric nitrogen gas, N2, into ammonia, or N3. It's called nitrogen fixation. And sometimes they cooperate, they work with one another and share these different resources because they can only do specific things on their own. So for example, here you see photosynthetic cells of an organism, of a prokaryote, and they exchange nutrients with nitrogen-fixing cells that provide the nitrogen for the photosynthetic cells. And you can see them here, this heterocyst, this combination of 2 photosynthetic cells aiding one another. And here there's other ones, such as in biofilms, prokaryotes covering surfaces of structures, including in your body, let's say in your intestines, where they cooperate with one another and exchange nutrients and help each other to survive. So there's a whole system of biology called molecular systematics that is trying to identify different prokaryotic strains and species, looking at the molecular biology, or their DNA, if you like, and trying to classify them. And it's some interesting results. There's even Craig Venter even built a yacht and traveled the globe for a year, collecting water samples from the oceans and trying to sequence new prokaryotes that we hadn't discovered yet, and he found thousands. And this is a new way that we're identifying different subdivisions or clades within the prokaryotic division. So here you can see, for example, this domain is called bacteria, this is archaea, and this is eukarya, this is you, that little branch there, all from some universal single-celled ancestor. When we say bacteria, we're referring to these, but prokaryotes are not all bacteria. Again, some are archaebacteria. Now, bacteria can be scattered among all different kinds, and a lot of times they're classified by what type of nutrition they use. We've talked about gram-positive before, and Proteobacteria are another group of bacteria here. These are just giving you some examples of them. For example, rhizobium here, this is involved in nutrient uptake in plant roots. So it assists or aids in the uptake of nutrients. And then there's other examples here. Helicobacter pylori, that's H. pylori. It lives in the intestines or the stomach of affected individuals and largely goes unnoticed but can cause problems and has actually been tied to stomach cancer. There's other ones here, cyanobacteria, gram-positives, and so on. Here's streptomyces. This causes strep throat, but is also used to... This does not cause strep throat. It's used to create antibiotics against strep throat. Here's chlamydia, one of your most known, because of the disease that it causes, of course, among others. This is just showing you the differences between bacteria and archaea, as well as eukarya, but mostly in here. A lot of this information you don't understand or know yet, and we will get to that. But archaea does share a lot of stuff with bacteria. Again, both prokaryotes. So extremophiles, as we showed you, some live in very hot environment. Here we have halophiles. They live in very saline or salty environments. There's methanogens. They live in swamps and marshes that produce lots of methane, and they love living in a high methane environment. All different kinds. There are tons and tons of prokaryotes. They're super important for survival and for the existence of our biosphere, all the living things. Everything is based off of prokaryotic life. So they play a really major role, including that of recycling of chemical elements between living and non-living sources, such as decomposers. When an individual dies or passes away, they need to get recycled. So breaking down corpses, dead vegetation, and various other waste products. In the soil, we have decomposers that make the nutrients usable again for plants to grow. Nitrogen-fixing prokaryotes, we already mentioned previously, they add nitrogen to the environment from the gases. They add nitrogen to the soil in the form of ammonia. And here you see a mutualistic relationship. It's a type of symbiotic relationship where both benefit. This fish has bacteria that live on it, but sometimes there's also commensalism, where one of them benefits and the other one is not harmed, but it is not benefited. Some live inside others. These are we call parasites, and some, including the parasites, they also live in humans, but there's also some that have positive effects. Here we see the Lyme disease in a human. They're being transmitted by a tick, of course, and some cause this damage or problems by releasing toxins internally or externally. And they can be used also as bioterrorism, although we don't see too much of this yet. We can use them in bioremediation, which basically means fixing or cleaning up the environment to remove pollutants. There are bacteria that can chew up oil, for example, in oil spills and recycle it, get rid of it. That was used in the most recent oil spill. They're used in the synthesis of vitamins, such as in vitamin B12. For pills, we synthesize that in bacteria. Insulin is synthesized in E. coli for people that are diabetic. The E. coli make it for them, and then they bottle it. All different kinds of ways. And we finish.